right, I think it's 9:30, so I'll go ahead and get started. Good morning. Thanks to all of you for being here for the first session on the last day. My name is Szarmach, and I'm a PhD student at Penn State. And today I'm excited to be talking to you about the results of one of my dissertation projects, exploring migration variation in myrtle warblers breeding in northwestern North America. So a wide variety of animals undergo seasonal migration, moving annually between a breeding and a non-breeding range. And in many of these animals, migration is an adaptation to seasonality and allows individuals to breed in areas where they would not be able to survive for part of the year. And a large body of work has shown for many taxa, aspects of migratory behavior has a genetic basis. This has been especially well studied in small songbirds, where crossbreeding experiments have shown traits like migratory direction are heritable. And using a variety of methods, we've learned that there's really a range of within-species variation in the migration routes that individuals take. So in some species, individuals from different populations all follow similar routes to their non-breeding grounds. But in others, different subspecies or populations follow different migration routes and winter in different areas. And new migration routes have also evolved over recent time, such as in the Eurasian blackcap here, where just within the last 50 years, some birds began wintering in Great Britain. And while we're increasingly able to characterize these migration routes, especially using tracking technology, this variation remains unknown for most species, and why alternate routes do or don't evolve remains an open question. One hypothesis for why some alternate migration routes might evolve is that migrating to a closer wintering area could reduce the energetic cost of migration and provide a fitness advantage for birds that are breeding in high latitudes. So for example, breeding in Alaska might result in a prohibitively long migration for some birds, but if birds begin wintering in California, a shorter migration route might allow for range expansion to these higher latitudes. And this might be the case for the parulid warblers, which I study, where species that migrate farther tend to have smaller ranges that often don't fill the full extent of available habitat. But there are a few warbler species with ranges that extend from the Atlantic coast all the way to Alaska, which raises the question of whether these species employ different migratory strategies that allow them to breed far from their core wintering area. And one of these few eastern parulid warbler species that has a breeding range extending all the way from the Atlantic to Alaska is the myrtle yellow-rumped warbler. And myrtle warblers have an interesting wintering range that actually has two disjunct concentrations. So their main wintering area occurs along the Gulf and the Atlantic coasts, but myrtle warblers have also been found wintering on the Pacific coast, particularly in California. So this raises the question, do myrtle warblers breeding in Alaska and northwestern North America follow an alternate migration route to the Pacific coast? The myrtle warblers wintering in California were first described in the late 1800s by Richard McGregor, who proposed that they were a new subspecies that he called Dendroica coronata hooveri. And the reason for this was he measured longer wings and tails in the birds wintering in California compared to those that were in the east. And in this first description, McGregor proposed that these California birds bred in Alaska and British Columbia. We currently don't consider hooveri to be a true subspecies today, but more modern studies of myrtle warblers in Alaska have found that these birds do have significantly longer wings and tails, similar to the measurements that were first described in California. So while these morphological similarities provide an indirect link between the California wintering ground and the Alaskan breeding ground, direct tracking of the migration routes of these birds would provide more concrete evidence to see whether these high latitude birds take a shorter migration route to winter in California. So that's where my project comes in. I deployed multi-sensor geolocators on 30 myrtle warblers breeding in Anchorage, Alaska. And traditionally, geolocators measure light levels that are then used to infer geographic locations. But our tags also included a barometer, so we could use a newer method that allows for more fine-scale location estimation based on atmospheric pressure. And birds with geolocators need to be recaptured to retrieve the data. So after a year, we returned to Anchorage and found that 6 of our tagged birds had returned, which is a pretty typical return rate for these kinds of studies. To analyze the data, we use 2 different methods. Using the light level data, locations are estimated based on the timing of sunrise and sunset. However, this method has some drawbacks. First, light level data can be affected by shading, especially in birds like warblers that spend a lot of time in foliage. And secondly, you can't estimate latitude during the equinox using this method because the day length is the same all over the world. And since fall migration in many species overlaps with the equinox, this really limits our ability to track movements over the whole year. So a more recently developed method is to track migration using atmospheric pressure. This is a plot of pressure measured by one of our geolocators over the whole year. And you can see these sharp drops in pressure, and that is when the bird is taking migratory flights at a higher altitude. So from these data, we can see every single day that that bird was migrating. To then infer the locations from this data, we use both the spatial and the temporal variation in pressure. So first, for every stationary period in between our migratory flights, the pressure data is used to identify all possible locations just based on the ground elevation. Then we can narrow down these elevations, or these locations, by comparing the changes in pressure over time measured by the geolocator to historical pressure data. And together, this allows us to produce a likelihood map for the bird's location at a given time. To do this, we used a pretty new R package called GeoPressure R that was developed by Raphael Nussbaumer. So before I show you the results of the analysis, I'll just remind you that based on our hypothesis, we expected that the birds breeding in Alaska are going to migrate along a shorter migration route to California. And what we actually found was completely the opposite. So all 6 of our birds actually migrated all the way to the Gulf Coast. So each of these maps shows the track for one bird based on our pressure data. The size of the point is how long the bird stayed at that location, and the color is just showing the time of year. So we actually found that rather than migrating to the closest wintering area, these Alaska breeding birds took a long route to get to the main wintering concentration on the Gulf Coast. Using the pressure data really allowed us to track these birds at a much higher resolution. We could estimate the timing of every flight and the location of every stopover throughout the full year. And the pressure data also let us identify much narrower regions where the birds were wintering. So on the right, the blue polygon is showing our likely wintering area based on pressure, and the red dots are showing the locations from light data over the same time period. And we can see there's a much higher latitudinal error in particular using the light. And then one last fun advantage of using this pressure data is that we could also characterize flight altitude over the whole year. And this is unknown for most migratory birds. So overall, the pressure data really improved our migration tracks and also gave us some additional information that you can't get from light. So while all of our geolocator tracks showed the same pattern of these birds migrating to the Gulf Coast, 6 is obviously not a very big sample size. So we also used stable hydrogen isotopes to estimate the wintering areas for an additional 167 birds from across this northwestern breeding range. So just for a quick primer on isotopes, because the ratio of protium to deuterium in precipitation varies across the landscape, the isotope ratio that we measure in a feather reflects the ratio of the area where it was grown. So comparing the precipitation hydrogen isotopes between the 2 wintering areas for our birds, we expect that a feather grown on the western wintering range is going to have a lower isotope ratio compared to a feather grown on the Gulf Coast. So for this isotope analysis, we used feathers that were collected during the breeding season in Anchorage, northern British Columbia, and Alberta. And because of the pattern of molt and myrtle warblers, we can collect feathers that were grown on the previous wintering ground. After analyzing the isotopes in these feathers, we found a significant relationship between hydrogen isotopes in feathers grown on the wintering ground and the longitude of the breeding ground where the feather was collected. So the feathers from birds breeding in Alaska tended to have lower isotope ratios compared to higher values in BC and Alberta. For each of these feather samples, I created a probability density map using the R package AssigNR, where our darker colors represent locations where the feather had the highest likelihood of being grown. And from these maps, we can see that feathers with higher isotope ratios are more likely to be grown further south, while those with the lowest include the West Coast in the region of high probability. You can see from this bottom plot that the Midwest, Appalachia, and New England are also included in the high likelihood areas, but these are much less common wintering areas for myrtle warblers. So in the maps, the green polygon contains the full wintering range, and the blue outline surrounds our most common wintering concentrations according to eBird abundance data. We performed an odds ratio test in AssigNR to determine which samples were more likely to originate from the West Coast rather than our eastern wintering concentration. So points below that dotted blue line are samples that had a higher probability of being grown on the Pacific coast. So while we can't fully rule out that these birds may have wintered in the Northeast, given that wintering myrtles are a lot more rare in that area, it seems likely that a small subset of our birds may be wintering on the Pacific coast. So how do these results fit into the range of intraspecific variation in migration routes that I mentioned at the beginning of the talk? One really interesting thing is that the routes that our tagged birds took is highly similar to routes tracked in another parulid warbler, the blackpoll. And for other species, such as the Swainson's thrush, it's been hypothesized that migration routes might retrace paths of postglacial expansion, and this similarity in routes between the warblers might suggest that they're following historical expansion of the boreal forest. However, the myrtle warblers don't follow exactly the same pattern as the blackpoll because we also found that evidence from isotopes that a small subset might be breeding on the Pacific coast. And this is more similar to some systems like the Eurasian blackcap, where you have a small proportion of birds in a population that are following an alternate route. So our findings lead to a number of follow-up questions. For example, are there other breeding areas where a higher proportion of birds are migrating to the Pacific? And then for those birds that do migrate to the Pacific, instead of taking this much more common route to the Gulf Coast, are there adaptive benefits for them to do so? So to summarize, we found using the geolocator data that all of our geolocator tracked birds migrated to the Gulf Coast, which was much farther than we expected them to go. But using stable hydrogen isotopes, we see that a small subset of birds may be taking this alternate route to the Pacific. And then finally, using this new pressure geolocation method, we were really able to better characterize migration routes and get additional information that we couldn't get with light alone. So with that, I'd like to thank everyone that made this project possible. And also, we're planning to post a preprint of this work soon. So if you're interested, feel free to email me, and I can send you a link once it's up. So with that, thank you for your attention, and I'm happy to answer questions if we have time. Thanks.
Okay, I'll go ahead and get started. My name is Nina Overgaard Therkildsen, and I'm going to talk about how the increasing access to increasingly cheap sequencing data is really expanding the toolbox that we have available for population assignment, especially of weakly differentiated populations. So this is work that was primarily led by my postdoc, Ryan Frankowiak, who happens to currently be on the job market. So if anyone has leads, let us know. But genetic assignment test is basically about identifying the population of origin of a sampled individual. So genetic assignment is widely used in conservation, evolution, and ecological applications, typically to look at when organisms move away from their breeding grounds. So for example, to track rates and patterns of dispersal, or look at patterns of distribution patterns or connectivity across space and time. So for example, complementary to what we saw in the previous talk, that could be figuring out which overwintering grounds are used by different breeding populations of migratory birds. It could be to identify long-distance dispersors or on something like bear populations, but probably the most extensive use of genetic assignment tests is to look at stock composition in mixtures of fish. So where different fish stocks come together at certain times of the year to estimate the relative abundance of different populations, so we can estimate fisheries impacts on different groups. And one species for which there's been strong interest in developing genetic assignment tools is American shad. So this species is distributed natively all along the east coast of North America, and it's an anadromous species, so it breeds in rivers, but goes offshore during much of its adult life. And it used to be extremely abundant on the east coast, forming the basis for some of the largest fisheries there. But unfortunately, it's been just completely decimated in many populations. And there have been a lot of efforts to restore habitat in rivers, but it turns out that one of the biggest threats to these fish probably is that they get caught out in the ocean in the marine stage of their life. So there's been strong interest in being then able to identify where fish that get caught in a bycatch in these marine fisheries, which rivers do they come from, so that fishing effort can be directed away from those areas where the most imperiled populations are getting caught. Unfortunately, early efforts to try to develop genetic assignment tools for shad were met with quite limited success. So there was an extensive study that screened a panel of 13 microsatellites across 33 rivers. And they found a good amount of structure, but most of the groupings were in Canadian rivers. And within US waters, which is where the management interest is most intense, only 2 major groupings were found. And a recent effort to try to use a similar microsatellite panel for genetic assignment in US waters found good assignment to those 2 groupings. But as you can see from the table here, the power to assign to individual rivers was pretty poor. So on average, only about 30% and all the way down to 4% of fish could be correctly assigned to the individual river. So that gives really limited usefulness sort of on the finer geographic scale. But fortunately, with the huge drop in sequencing prices, we're getting access to a lot more information with now being able to screen the whole genome. And in my lab, we've been really interested in exploring how we can get the most bang for our buck in terms of designing sequencing experiments for these kind of conservation projects where we often have quite limited budgets. And what we found through a number of different simulation studies is that typically, or for a lot of population genetic parameters, we get a lot more information from sequencing many individuals at low coverage rather than sequencing few individuals to high coverage that you typically do if you need good genotype calls. So this is the approach we took. This low coverage whole genome sequencing is what we used to start exploring the geographic patterns in shad. And if we're just looking at a PCA here of the genome-wide variation, we see that there's quite a bit of geographic structure. And in particular, we were really excited to see that in the US region, where the previous microsatellite analysis had only been able to identify 2 major groupings here marked in green and blue, we see actually separation between most of our sampled rivers. So there's a lot more to work with here. And this is just a genome-wide pattern. If we then look at Manhattan plots here, where on the y-axis for each of these panels, we're looking at FST, so the degree of genetic differentiation between neighboring populations for SNPs across the genome. We see that there's a lot of heterogeneity in the amount of differentiation, and we see several distinct peaks, and of course also something that looks like an inversion in the genome. So it's clear that if we're going to be designing assignment panels, so we're going to be selecting markers to develop assignment panels, we get a lot more information if we pick a SNP that's located within one of these peaks where allele frequencies are really strong between the groups that we're trying to distinguish, compared to we just pick random SNPs that reflect the background amount of genetic differentiation, where there's much less allele frequency difference, so much less information to be able to distinguish our populations. And most of the prior microsatellite markers were most likely just from this background level, where there's very limited differentiation between groups. So already just looking at that is one example of how whole genome, being able to screen the whole genome is improving our ability to do assignment, because we can go and specifically pick markers from the most highly differentiated regions. So we did some initial simulation assessment to evaluate just how much they could improve our assignment power, and this assessment suggested that with as few as 100 SNPs, we would be able to get greater than 90% assignment accuracy to each of our populations. So we went ahead and developed a cost-effective genotyping panel using a GT-seq, so a highly multiplexed amplicon sequencing approach. And we started out by screening almost 600 of our markers for various technical and unfortunate reasons. We had a huge dropout. So for this first panel here, we ended up only getting good genotype data for about 100 of them. But when we then went and used this panel to genotype 20 new individuals from each of our populations. So these were individuals not used to select the SNPs, so we avoid high grading bias, but in a new set of test individuals, we saw that just with these 100 markers, we were able to, for some of the locations, we were able to get, showing here the proportion of individuals that got correctly assigned. For some locations, we got over 90%. Other locations, it wasn't as great, but still substantially better than what could be done with the microsatellite markers. And if we group some of the individual populations into reporting groups, it's looking even better. But quite frankly, we were really disappointed with this because it wasn't nearly as good as our simulations had predicted. So we're now going back and designing a new panel with more markers that I think we're still waiting to get the sequencing data back to see how much that improves the performance. But in general, developing these kind of panels is quite costly and time-consuming. And also, because we are selecting SNPs to compare specific populations, and then we make an investment to buy these primers and work to optimize them to place nice together. We make a lot of investment in specific comparisons in a panel that might have less usefulness if we suddenly want to compare different populations. But most importantly, I think, is also that, of course, a small panel of markers will not capture the full amount of variation. So we see that here very clearly when we look at a PCA based on these 107 markers we were able to genotype in our reference individuals compared to the PCA of the full genome data, the low coverage data, where we can just see there's so much more resolution when we're able to leverage the full genome data. But unfortunately, sort of the motivation for developing the panel was that until very recently, all the genetic assignment software required called genotypes. So we couldn't use that directly with the low coverage data. But quite recently, Matt Six and Eric Anderson published this new method that's developed to specifically leverage genotype likelihoods directly from low coverage data. So it sidesteps the need to call genotypes. So to be able to test that in our data, we started out by just boosting our sequencing coverage a little bit, up to about 3.5x for each of the 30 individuals per location. So still quite low coverage, but just to have a little bit more information to work with. And then we looked across the genome. And if we're just taking a look at the assignment results, so in this matrix here, we have, in the columns, we have the populations or individuals originated from, and in the rows, we have counts of which populations they get assigned to. So we can see here from the centering on the diagonal that we have pretty good assignment, 100 and 100% correct assignment to most of our populations. In this first pass, there were some geographic regions where quite a large proportion of individuals were assigning to neighboring rivers. But it turns out that a lot of that was driven by slight differences in the number of individuals and the sequencing depth, differences between our different reference populations, which is known to bias this method. So when we downsampled our reference data to have more even effective sample size between the different locations, that actually cleaned things up quite a bit. So we can see before we had, for example, from this Kennebec River, we had 9 of the 30 individuals assigned to the neighboring Merrimack. But after we standardized the sample sizes, that's only down to 3. So we're getting better performance this way. But what I think is especially exciting is that this previous work has shown that this near-perfect assignment accuracy is maintained even when we downsample test individuals down to a super low level, like 0.01x. So basically, because as long as we have sufficient or sort of decent depth on our reference samples, because we have data across millions of sites, we only need a relatively small proportion of those sites to have read data for to get enough assignment power. So this means that for something like Schlatter that has a genome size of about 1 GB, we only need about 10 megabases of sequencing data, which costs less than 10 cents, so hardly anything really. So of course there are the sample preparation costs, but at least in our hands, those are kind of similar to what it cost us to genotype a sample with this GT-seq. So of course there are initial investment costs for each of these methods. With the GT-seq, we have to buy all the specific target primers. With the low coverage approach, we have to generate some reference data at slightly higher coverage. But the running cost for sort of routine screening of a test sample is very comparable between the two. So I think, of course, there are pros and cons to each method, but I think it's really exciting to see that this low coverage approach can actually be implemented at kind of similar cost, because I think the big pros of that approach is especially the flexibility that we are not committing to comparison of any specific populations, and the sample preparation is actually much simpler to just prepare a shotgun library. And then that, at least in this case here, we seem to get better accuracy for weakly differentiated populations. So there'll be, of course, practical constraints about sort of computing power and setup where that won't make it ideal for all situations, but I think it has a lot of potential moving forward. So with that, I'd just like to thank other collaborators and our funding sources, and thanks for listening. I think we have time for 1 or 2 questions. Yeah?
How do you decide the confidence assignment when you really want to?
Yeah, so that's a big also issue with this method, is that it's so new that there still isn't really established, as you know, the method. So for this, what I was showing was actually just without any filter on posterior probability, just best assignment for everything. But certainly it can be refined with various screening for levels of confidence in the assignment.
Not sure. Go ahead. Good morning. My name is Schlatter. I'm a PhD student in the Boston lab at Boston University. And I'm going to talk to you today about connecting dispersal and spatial genetic structure in Nyanggobe, Elecatinas lori. So we're in a migration session. When we're thinking about ways that organisms move around, one important process in the marine environment is larval dispersal. So this is a sort of cartoon of that in a clownfish. But for many marine taxa, they have this bipartite life cycle. And it starts with the dispersive larval phase. So juveniles spend their first days, weeks, or months in open water. And they can travel often long distances. So in some species, hundreds or even thousands of kilometers. And at the end of this phase, if they survive, they can recruit back to adult habitat. And in a lot of species, they then remain relatively site-attached. So larval dispersal can be sort of the main source of exchange between populations. And because of the potential for long-distance dispersal and the idea that larvae kind of just ride around on the currents, it was once thought that marine populations were kind of largely panmictic. But in the last few decades, we've learned that they often exhibit measurable spatial genetic structure. So a major question is, can we understand the evolutionary process of how dispersal leads to spatial genetic structure in these systems? And we have theory about how it works in sort of an idealized case, things like the island model and isolation by distance. But of course, the reality is more complex. It involves sort of complicated spatial habitats, configurations, and oceanographic regimes, which could change over evolutionary timescales. So my goal is to work on answering this question in practice. And I'm using the Nyanggobe, Elecatinas lori, as a model system. And they're a coral reef fish. They live as adults in these yellow tube sponges that you can see on the left. They're these cylindrical things they live inside. And they lay their eggs in the sponges, and then the larvae disperse. They develop pelagically for about a month. And then they recruit. In the middle there is a picture of a new settler on the outside of a sponge. It's kind of hard to see, but it's there. And because they live on the Mesoamerican Barrier Reef, so that's a sort of spatially complex habitat. There's the linear barrier reef that's mostly continuous with some breaks, you can see on the top. And then these circular atolls that are at various distances from the barrier reef and from each other. They're a relatively easy species to find because they live in these tube sponges, so that facilitates fieldwork with them. And thanks to previous work in our lab, we have really great data sets on both their dispersal and spatial genetic structure. So first, we have very well-resolved estimates of dispersal patterns. By sampling a massive number of adults and newly settling juveniles in a 40-kilometer stretch of the barrier reef, and then doing parentage analysis, Deloya et al. Were able to estimate the dispersal kernel, and that's what you see at the bottom left there. It's a probability distribution of dispersal distances. And most larvae stay very close to home, and the farthest dispersal events that were measured were in the 10 to 20-kilometer range. Second, we have information on their spatial genetic structure. So over the area shown here on the right, which is about 200 kilometers north to south, Deloya et al. Sampled adults from each of the 34 sites pictured. And they uncovered this hierarchical genetic structure. So the broadest scale of that is what's shown in the figure here, and they found 3 distinct genetic clusters. So the Barrier Reef and Turnef clustered together in blue, Lighthouse Atoll in yellow, and Glover's Atoll in orange. And they also found finer scale structure within the Barrier Turnef cluster, and also a pattern of isolation by distance along the linear reef. And I'll show you both of those later. So we have these really nice empirical measurements of dispersal and spatial genetic structure. And the question is, can we link them? And what can this tell us about the process of how dispersal affects evolution? So I'm going to approach this question using an evolutionary simulation. I'll try to predict the spatial genetic structure of Elori, given what we know about the dispersal kernel and the contemporary seascape. And notably, this means I'm not using any information about the oceanography or about how the seascape has changed over time, both of which could be relevant to dispersal and therefore to evolution. So it's not immediately clear whether this should work. So to get from a dispersal kernel to a prediction of spatial genetic structure, I'm using an evolutionary simulation that's implemented in SLIM. So it's an agent-based model, and it simulates the birth, dispersal, reproduction, and death of individuals over many generations, while keeping track of how genetic information is passed down with recombination, mutation, and mating. And the dispersal distances here are determined by the empirically measured kernel. It's spatially explicit, so I've given it a map of the barrier reef and atolls. And we've added life history, behavioral, and genetic information to the best of our knowledge in the system. So after many generations of simulation, we take samples from the simulated individuals according to the same sampling scheme used for the empirical data. So the same 34 sites and same number of individuals from each site. We repeat this many times to account for stochasticity among simulations. We filter the simulated genotypes according to the same criteria used for the empirical data so that they match. And then we use this simulated genetic data set to calculate pairwise FST, or genetic differentiation, among all 34 sites. And then we can compare the observed pairwise FST values to the simulated ones to see how well our prediction has worked. So the results I'm going to show you today are pretty preliminary and qualitative, and there are a lot more sophisticated methods of comparing simulated and observed data that I'd like to try, and metrics other than FST. But what I have today is kind of meant to answer the question, does this method work? So here's what we find. On the left, and just for reference, is this map again of the broad-scale genetic structure and the 3 clusters. And the plot shows you the empirical data first. So on the x-axis is pairwise FST between any 2 sites, and the distributions are colored according to where the 2 sites are located. So the solid colors represent pairs from the same group. So for instance, the yellow distribution is FST between pairs of sites that both belong to the Lighthouse cluster. And the stripes represent pairs from different groups. So Barrier Glovers is on the left, Glover's Lighthouse is in the middle, and Barrier Lighthouse is on the right with the highest FST values. So we can see from this, as expected, that sites within a group are more similar to each other than sites in different groups. And now we can compare this to what we get from the simulation. And as you can see, they show the same broad pattern. So differentiation is lower within groups than between groups. And there are some differences between these plots, which I'll talk about more in a minute. On a finer spatial scale, the same pattern is present empirically. So we have the 3 genetic subgroups, North Barrier, Central Barrier, and Turnef in the map. And you can see in the empirical data that differentiation is again stronger between them, so the stripe distributions, than it is within groups, the solid colors. But at this scale, the empirical structure is not reproduced by the simulation. In this case, the highest FST values are all for site pairs where at least one site belongs to the northern cluster, so all these flat, long distributions. So there's something going on in the simulation at this level that's not quite right. And finally, we can zoom in a little further and just look at the linear barrier reef. And the empirical data show this clear pattern of isolation by distance. Each point here is a pair of sites, and the x-axis is the Cartesian distance between them, and y is the FST. And so as physical distance between sites increases, so does their genetic differentiation. And we see the same qualitative pattern in our simulated data. So at this scale, we also have sort of qualitative agreement between the two. So we built a simulation that can predict qualitative patterns of FST based on the known dispersal kernel. Not perfectly, but we're sort of reproducing most of the general patterns. And notably, we're doing it using the contemporary seascape without needing to know anything about oceanography or about how the seascape has changed over time. And this is worth paying attention to because it's often thought that we really need to understand oceanography to understand dispersal. And here we're doing a pretty good job without. But there's one major quantitative difference between the empirical data and our simulations, and that's the scale of FST. So returning to these plots for a moment, you'll notice, or you might have seen before, that although the qualitative patterns are very similar, the scales differ by an order of magnitude. So the simulated FST values are much too large. So we can ask why this would be. And this is a neutral model. We're not thinking about selection, and so the processes that affect FST here are essentially migration and drift. And on the left here is the formula for FST in the classic island model, which doesn't apply here directly. Our system doesn't meet the assumptions, but it illustrates the general way that FST, migration, and drift are related. So if our simulated FST values are too high, then one reason could be because our estimate of NE, or effective population size, is too low, so drift is too strong. And in fact, the population size in this model is too low. We estimate there are on the order of 500,000 individuals in the geographic range covered by the simulation. But for computational speed, I only simulated about 50,000. So one of my next steps is to increase the efficiency of the simulation so I can try and address this issue. Second, and these aren't mutually exclusive, it's possible that migration M is too weak in our simulation. And in particular, that we're not modeling enough long-distance dispersal. The empirical kernel is really well resolved at short distances, but it was fit using an exponential function, which means that the tail is pretty thin. And there's evidence in other species that dispersal kernels are often fat-tailed. And in this species, Deloya et al. Have recently identified additional long-distance dispersal events using the same data through population assignment. And so changing the kernel a little bit also seems like a plausible hypothesis. So my first step moving forward is to think about these two things to try and resolve this discrepancy in scale. And then if we can do that successfully, then I plan to take a look at remaining ways that the observed and simulated data differ, and use these to test hypotheses about what processes are important in the relationship between dispersal and spatial genetic structure. I'll offer one current discrepancy as an illustration, although these could change after we fix the scale issue. So our current simulations predict that Glover's and Lighthouse should be the most strongly differentiated of the three broad-scale genetic clusters. That's the distribution on the right with the largest FST in the simulated plot. But we see that those are switched empirically. Glover's and Lighthouse are more similar to each other in the empirical data than Lighthouse is to barrier turnip. And there's a good ecological reason that we might expect this to be the case. So there's a strong current that we know about that moves northwards from Glover's to Lighthouse, which could account for these populations being more connected than we would otherwise expect. So this might be a place in the seascape where we do need some information about oceanographic distance in order to make good predictions about spatial genetic structure. So altogether, we've been able to build a simulation model that sort of qualitatively predicts patterns of spatial genetic structure without using history or oceanography. And if anyone has ideas about things to think about as I try to resolve the issue of FST, I'd be really interested to chat. And assuming that we can overcome that, I think there will be sort of a lot of interesting work to do looking with high resolution where in the seascape the simulated and observed data agree and where they differ, and asking what that can tell us about sort of how the underlying process of dispersal leads to evolution. So I want to acknowledge the many people who worked over multiple years to collect the field data that went into this project, my colleagues in the Boston lab, my PhD committee, and folks in the biology department for helpful discussion and feedback and the funding sources. And thank you for listening. Yeah.
Thank you. That was a really lovely talk.
Thank you.
I'm curious, this is such a short disperser and such a short distance. Do you think that really oceanography is just going to be a much bigger player in the longer dispersing species?
I don't know that we necessarily have evidence that that's the case, and I think there's scales at which oceanography is important too. Like there's sort of local scale eddies that can be really important, and I think we're learning more that those can be really make a difference as far as sort of retention. So it's hard to say, but I think that's a good question.
So my name is Louis Bubrig. I am a PhD candidate at the University of Virginia in the Gibson lab. And today I'll be talking about dispersal as a strategy to avoid the late stages of an epidemic. So I don't think I have to convince this room that dispersal is ubiquitous. It is highly dramatic and seemingly very costly. And so when we're thinking about why an organism would choose to disperse, a lot of the theoretical explanations come back to this concept of habitat deterioration, which is powerful enough to drive the evolution of dispersal. And so very basically, over time, the idea is that habitats can become more unsuitable, and they may become so unsuitable that the benefits of dispersing outweigh the costs of staying put. And so if we want to test these theoretical explanations, we need to ground the concept of habitat deterioration into the actual ecological forces that could drive it. And one really nice candidate for the drivers of habitat deterioration are antagonistic interspecific interactions. And in particular today, I'm going to focus on parasites. And for simplicity, I'm going to keep saying parasites, but parasites, pathogens, disease are going to be kind of under this umbrella. And these are really exciting because they're universal, so every organism on Earth has parasites. Their spread is inherently temporal, and so that habitat deterioration can occur over time. And good thing for us, the progression of that is fairly easily quantifiable, all things considered. Now, one really intriguing thing about the temporal nature of parasites is this idea that you can start with a small inoculum. So you can have a population, you can either introduce a single infected individual, or perhaps spores or some kind of environmental stage will come in, and then that will over time ramp up to an epidemic. And so that kind of provides an interesting opportunity for hosts to be a little bit clever. And instead of waiting until it gets to full-blown epidemic levels and then deciding to disperse, if they can somehow detect it somewhere closer to the small inoculum stage, then they might have the opportunity to disperse preemptively and actually just avoid the late stages of an epidemic entirely. So that's kind of my big hypothesis here, is that hosts will attempt to pull this off. And so the way that I'm going to be testing this is basically just breaking this down into two easily testable predictions. So in the presence of parasites, my first prediction is that hosts should invest in early dispersal in comparison to the timeline of the epidemic. And then my second prediction is just that those early dispersers should benefit from that by being healthier than the dispersers that decided to leave late. So the system that I'm going to be studying this in is the host species Cinorhabditis elegans. Here's what they look like, little microscopic worms. Here's a population. And it's parasite Nematocida parisiae. So this is what it looks like together. These bright spots are a fluorescent probe that I have tagged them with. And these things have been probably co-evolving for millions of years. They're found together all the time in nature, all across the world, all across C. elegans range. And C. elegans is also a really natural system to study dispersal in because C. elegans has so many adaptations to facilitate dispersal based on its habitat, which I'll show in a little bit. And also it's C. elegans, so it's very tractable in the lab. So the way that this works in nature is that C. elegans has a dedicated dispersal stage called the Dower stage, which is loaded with all these adaptations to facilitate dispersal. And so they will ride on a little carrier species like a snail or a bug. They're adapted to find those creatures. They're adapted to hang on to them until that, that carrier organism finds something like a new bacterial bloom that's on a rotting piece of fruit, for instance. It'll drop off those dispersers. Those dispersers will start reproducing in the normal life cycle, and that population will get denser and denser. And then generally, as it gets denser, it's more likely to produce more dispersers that will find another carrier and continue. So we already know that dispersal in C. elegans, there's a density-dependent component. We'll kind of stick a pin in that for later. But I essentially just want to take this and just bring it into the lab where I can just quantify and set up these really controlled epidemics and be able to track them in real time. So my methods are basically to just start a population much as they would in nature with about 20 individuals. That's about the range of how many new colonizers we expect. Half of them are going to get a pretty low dose of Nematocida parisiae. And then I'm just going to track this over time points that represent kind of the full range of epidemic progress. And so in particular, the first 5 days after introduction of that parasite, just because that's the time, I'm really interested in the shifts that happen early in the epidemic. And then I'll also capture these kind of later time points when the epidemic is really, really bad. And so basically, for my first prediction that says more dispersers should be produced when exposed, I can just count the number of dispersers that have accumulated up to that time point to basically see when does investment in dispersal start, when does it peak, and what's that magnitude. And then for my second prediction that early dispersers are healthier, I can tag active infections with fluorescent probes. I can use fluorescent microscopy to determine the epidemic progress up until that point. And I just want to point out that all of this work was done alongside former Gibson Lab undergraduate Sarah Talley. So for my first prediction, which is that hosts should invest in early dispersal, I can just check how many dowers have accumulated up to that point. So here's my kind of example populations. I'll filter out the dowers, I'll quantify them. And as this shows, and as I mentioned before, the sort of null expectation in the control is that as the populations get more dense day after day, we should expect to see more dowers. And that's exactly what we see, right? So as time progresses on the x-axis, we see that the number of dowers pretty steadily increases. And then if we look at our exposed, we see that there are more dowers almost completely across the board, except that late time point, and also in the exposed treatment, that peak is shifted about 5 days earlier. And so this is pretty dramatic. Keep in mind we're on a log scale here, so this is a pretty big bump to dispersal. So my question then was, how is C. elegans actually doing this? Because that might be able to give us some information on how early and what they're actually detecting in the environment and what they're shifting in their life cycle. So one option is that exposure could induce dispersal directly. And what I mean by that is, again, we already know that there's a couple of different cues that trigger dispersal in C. elegans like density. But if I do a small little controlled assay where I keep all of that exactly the same, the only difference is that I put these infective spores in the environment, do they make more dispersers? And the answer is no, not really. So the spores themselves being in the environment is not what directly triggers dispersal. However, I'm clearly seeing that they're producing more dispersers. So the other option is that they could be inducing dispersal indirectly. Basically, they could affect some Q for instance density, and then that in turn is actually leading to more dispersal. So my kind of like back of the napkin math, if we are looking at density, is that they would have to, in the exposed condition, boost their density quite a lot in order to pull this off, and that would have to come very early on in the epidemic to correspond with that timeline. So here again, I'm just like looking at the number of worms in the population. And in the control, we see pretty typical, we're from those 20 initial individuals, we're getting up to about 150,000 individuals, and then it kind of plateaus there. And then in the exposed, they do this. So their population completely explodes. Notice the scale here, so we're reaching about 500,000. That's 3 to 4 times the size of the control population, and then that ultimately will crash below the control. But that timeline completely corresponds to that investment in dowers that we saw. So our interpretation of this is that exposure to N. parisiae is triggering an explosion in population growth that is then causing a larger investment in dispersal. So my second prediction is that early dispersers should be healthier than late dispersers. This is super easy to see visually. I can just show you. So those early dowers, they look like this. And you might not even be able to see, but in the top left worm here, at the very tip, there's a little bright spot. So this is a representative sample. Most dowers very early on are uninfected, and the ones that are, they might have like a tiny little infection. And then you check later, and they look like this, right? So almost all of them are infected, and when they are infected, it's completely through their entire gut. And so just quantitatively, right, we can see that the percentage of dowers infected, so day 3 is the first day that we see dowers at all, so we don't have any data for 1 or 2, because there are no dowers there. But the first day that we see dowers, they're fairly healthy, all things considered, and then that percentage shoots up. And also, if you look by the percent body area that's infected, you also see that increase over time. Now, something about C. elegans is that you kind of end up working with single genotypes at a time because they're so likely to self. And so all these previous results were done in a standard genotype N2. And so I just wanted to make sure that this was not some quirk of this particular genotype. And so I also pulled in a genotype called CB4856. This is considered like a more kind of wild version. It's pretty distant within the sort of strain phylogeny of C. elegans. And so this is going to allow me to just make sure that both of them are roughly doing the same thing, but also check for variation, because we do know that between genotypes within the species, there is variation in dispersal propensity and some of these other traits. So I'll just kind of like run through this pretty quickly. We saw more dowers in exposed conditions in N2, that's what I already showed. And CB, we see the same thing. In N2, we said that that corresponded to this increase in population growth in the exposed treatment. And when you look at CB4856, does the same thing. And if you remember, N2 did not have dower induction directly, meaning if I kept everything the same, adding those spores didn't actually make a difference. However, in CB4856, we also see that in addition to that population growth, they also seem to just directly have dower induction. So that's a factor of like 2 or 3 above the control. And what's pretty interesting and what might hint at how they're actually detecting the early stages of the epidemic is that if I deactivate those spores, so they're still in the environment, they're just no longer capable of infecting anybody, that pretty much goes away. So it seems like it's infection dependent, and then that's triggering something. So just to recap, my hypothesis was that hosts disperse to avoid late stages of an epidemic. And I found that there were more dispersers in exposed treatments, and that could be through 2 non-exclusive mechanisms. It could be through accelerating population growth in the exposed treatments, or it could be, or could be through direct induction. And I'm probing a little bit more into this mechanism, again, just to figure out exactly how early are they detecting this, and then how is that translated into shifts in either their development or fecundity or things like that. And then my second prediction was that early dispersers should be healthier than late dispersers. I found that they're far less likely to be infected, and also the ones that are infected have less severe infections early on. And I still don't have the microscopy data for the CB4856, so I'm going to do that just to kind of confirm. And then I've started an experimental evolution project where I basically have mixed populations of genotypes exposed to Nematoceta parisiae to see if that early dispersal can actually evolve in those situations. So with that, I'd just like to thank everybody in the Gibson lab and particular people who've helped on this project, funding sources, and I can take any questions. I am not sure. It's a little bit hard. One thing that's tricky with C. elegans is that sometimes if you look at an individual worm and you look at like their fecundity over time, it doesn't map very cleanly onto the population growth. It's a huge, huge increase. So I think it must have to do with, clutch size or the percentage of worms that are surviving that clutch, maybe through like provisioning or something. But yeah, it's a huge response. I'm hoping that if I probe deeper, I'll be able to find it. But yeah.
Hey, good talk. You talked about that shared history between the parasite and the host. Do you think that what you see here could be a direct artifact of that, and maybe you wouldn't see this if you tried to do the same experiment with a pathogen that we find worms with in the wild, but they don't have that evolutionary history with it?
That would be my prediction. I think that this is a signature of them interacting quite a bit. I haven't gotten exact numbers on like what, when you go out and sample C. elegans in the wild, what percentage of those populations have N. parisiae, but I'm led to believe that it's like fairly substantial, like you find it a lot. And so I think they're dealing with this all the time, and yeah, I think that this is the signature of that strategy having already evolved.
Have you checked for any particular metabolites that may be produced by your infectant that might be affecting C. elegans as well?
By the parasites? I have not personally, but I do know, so they're intracellular parasites of the gut. And so there's the potential either for them to be producing some metabolite or them to just be interfering with the host's own metabolism somehow, and then that leading to some chain of effects. So.
So can the dowel be infected? And if you have an idea of it, if so, if the idea of the proportion of infected dowel you have.
So the dowers, so this is an oral parasite, and dowers don't have like a functioning gut, so they've completely sealed off their gut. So what we imagine is happening is that they're sort of encapsulating whatever infection they had in the previous habitat and carrying it with them. So I don't think that dowers themselves, if they're uninfected going into the dower stage, I don't think that they can then be infected. So the ones that we're seeing very early on at those time points that are uninfected, they are basically in the clear until they get to wherever they're going, we think.